brings us to the next speaker, which is Giannis. His camera is already on. Good.
Can you see me and hear me?
Yes.
Yes. Okay.
We can hear you, we can see you. Oh, good. So our next speaker today is Dr. Ioannis Stringlis. He's an assistant professor of phytopathology at the Agricultural University of Athens since January 2023. He is working on plant-microbe-pathogens and pathogen interactions, mechanisms of induced systemic resistance in plants, interaction between iron deficiency and pathogens, and plant defense mechanisms activated in response to phytopathogens and beneficial microbes. Prior to his appointment as assistant professor, he has performed his PhD in the plant microbe interactions group at Utrecht University, Netherlands, under the supervision of Professor Corné Pietersen. During his PhD, he studied how plant roots respond to beneficial microbiota and activate systemic responses. After his PhD, he continued in the same group, initially as a postdoc and later as a junior group leader, where his research focused on the role of coumarins in plant microbe communication and the role of chemical and structural defenses in plant microbiome assembly and interaction with beneficial microbes. He has obtained his Bachelor in Science in Agricultural Biotechnology and his Master of Science in Molecular Phytopathology in the Agricultural University of Athens. So without further ado, Giannis, the floor is yours.
Okay, thank you for this really nice introduction. I will try now to see. Can you see my pointer? Yes, right? And then you can, let me see, I need to press this. Okay, so thanks for this really nice introduction and for giving me the chance to present you, first of all, the university where I work now. I will say also some things about the lab where I'm based. And also I will give you some highlights of the work I'm doing. I'm still affiliated with Utrecht because there are still ongoing projects there. So I will present you some information about this, but also the work I have started the last 1 year and a half, more or less, since I came back to Greece. So, okay, you can see my slide. So Agricultural University of Athens is based in Greece. So you might all know where Greece is in the east part of the Mediterranean, and actually the university is based in the capital, Athens. So if you have been to Athens, let me see, I'm using this, okay. So Athens is actually the city center of a bigger metropolitan area, okay, and that is starting from the seaside and ending up to some mountains that surround the city. And Athens is really known for the ancient antiquities that you can find there, like Parthenon and so on. So if you don't know Athens or Greece for these reasons, you might know Greece for some, let's say, culinary trademarks, like ouzo, feta, or gyros with this nice tzatziki sauce. So this is about where the university is. But now some things about the university where I work. So actually, I work at the Agricultural University of Athens. It was established by law back in the 1920s. And it's the third oldest university in Greece. So back in time, when the university was established, Athens was not in the size where it is nowadays. It used to be orchards and plain fields and so on. And here you see the main administration building where the rector is, but also where our lab is located. So the situation nowadays is a bit different from these orchards that I show you. So we have a lot of concrete, but hopefully our university is a small green oasis in the city center of Athens. So you can see here, if we zoom in, there are still experimental fields, orchards with different trees like olives, almonds, and so on that we use for experimentation. And you can see here the different buildings and facilities that are available. So I would like to tell you also some things about the main pillars of the research we do at the university. So it's an agricultural university, so we work a lot with plants, animals, animal feed, food production, environment, but also with microbes. And we study them using different techniques, or let's say by assessing them by different levels, let's say genetics, biotechnology, nutrition, management, economy, and disease prevention. And the aim is to increase yields, food security and quality, to increase innovation, but also sustainability in agricultural practices. So there are 14 departments in the university. The lab where I work is in the Department of Crop Science, but there are also different departments that are mostly working with animal science, agricultural engineering, food science and human nutrition, biotechnology, agricultural economics, and so on. As I already mentioned, there are 14 departments. Within these departments, there are 19 postgraduate degree programs, 8 PhD programs. All the activities of the university are based in 28 building complexes, where you can find 40 fully equipped labs. And then for teaching, there are 55 auditoriums and teaching rooms, and also 13 greenhouses for experiments. Additionally, out of the premises of the university, there are also experimental fields where we can apply, let's say, innovations or experimental findings that we develop in the lab out in, let's say, the real life situations. And here you can also see the agricultural museum that is also found in the premises of Agricultural University campus. We also have a vibrant student community. Every year we have more than 250 Erasmus students that come to our university for in different semesters, okay, to follow some courses that we give. We have more than 8,000 active students. There are gym and there is a really nice and fully renovated gym, but also different sports and cultural teams. And recently, we have established an innovation and technology transfer office that really helps us scientists transform our ideas into solutions for society. Now, some more things about the university, the lab where I work in. So this is the structure. We are 6 PIs working on different but also complementary topics, as you will see in the next slide. There are 9 PhD candidates, 3 research associates, 1 technician, 2 teaching staff members, and more than 20 bachelor and master students. Now, about the expertises that you can find in our group. So we have a plant virologist, Dr. Elisabeth Hadjivasiliou. As you can read here, she's working mostly on the diagnosis, identification of plant viruses, plant virus vector interactions, and evaluation of methods to control plant viruses and their vector insects. Professor Dimitris Giannis is working on biological integrated management of plant diseases, mycotoxogenic fungi and mycotoxins, but also on how to apply digital technologies and precision agriculture tools to improve remote diagnosis and monitoring of plant diseases. Then the head of the group is Professor Paplomatas. He's working on the diagnosis of soil-borne pathogens, biocontrol of plant pathogens, virulence genes of phytopathogens, but also application of RNAi technology to combat pathogens. Then another colleague of mine is Professor Aliki Dzima, that is working on a soil-borne pathogen, Verticillium dahliae, and she's studying the role of virulence gene in this pathogen and the role they have in the interaction with the host, but also epidemiology of olive anthracnose, but also molecular diagnosis of plant pathogenic fungi. And then we go a bit lower down, below the surface of the soil, and then it's Professor Sotiris Zamos, that's working with biological control of plant diseases, but also plant pathogen interactions. And he has a strong network with industry, but he has quite some projects with them. And then it's also me, and as Naomi really nicely explained, I'm working on plant-microbiome-pathogen interactions. I study mechanisms of ISR in plants. I also study the interaction between nutrient stresses and pathogens, but also how plants respond to both pathogen and beneficial microbes. So, Our lab is fully equipped, so there are 4 rooms for research. There are 2 rooms that we use for teaching, and then we also have a room for compounds and materials, but also the offices. It has facilities for plant and microbiological research. You can see here some of the things that we have. We also have microscopy facilities. We have a confocal laser, but also fluorescent microbe and microscopes. And then we also have greenhouses where we can perform experiments with both model and crop plants. There is a number of funding that is now currently running in the group. So there are some Horizon projects. Then there are some Erasmus or Marie Curie staff exchanges projects. I will elaborate a bit more on this because I'm also highly involved in this one, but also national funding, all related to plant pathology, plant microbe interactions, and how to use new technologies towards more sustainable agriculture. Let me see. Okay. Now, some more things about micro-SOS that I mentioned in the previous slide. Actually, this is a Marie Curie Staff Exchanges project. In case you're not familiar with that, this is a project that really facilitates interaction between academia and non-academia, but also knowledge exchange and transfer between different partners. And as part of that, we can also perform some nice research. So as you can see here, this Marie Curie is having Agricultural University of Athens as a representative as partner from Greece. The coordinator is the University of Malaga, Dr. Victor Carrion. Then we have partners from France, 2 companies, Equinao and Desangos, University of Tousa from Italy. And then we have also some partners from the Netherlands, Leiden University, NEO, but also a company called Weather Makers. And we also have Asian Institute of Technology as an associated partner from Thailand. So what the goal of this project is, apart from knowledge transfer, is to also find, let's say, to use microbes, as you can see from the title of the dramatic title, I would say, of the project, plants crying for microbial help in a changing world. So actually what we want to do, we want to find some microbes that we can then use as synthetic communities. And then we want to apply these microbes. And you can see here different work packages. And then we want to apply these microbes in greenhouse settings using tomato, and then see if these microbes can help the tomato plants deal with different biotic and abiotic stresses. Actually, our university, and I represent the university in this project, I lead it, is leader of work package 2. Then we also have work package 3, where we are mostly going to do data integration of all the things that we are going to collect from work package 2. And then we will try also to apply with the help of companies some of these findings in let's say, agricultural practices. So this was some, let's say, information about the university, about the lab where I work, about some of the projects we run. But I would like also to give you some highlights of my work. And my work is really centered around the plant defense system and the role it has in plant microbiome interactions. And as I already mentioned, there is a number of projects that I still run with my colleagues in Utrecht, because there is a number of PhD and postdocs that I co-supervise together with Corné Pieterse, Saskia van Wes, and Ronnie de Jonge. But also, I have some projects. I have a project actually that I have started now since I came back to Greece. So our projects are trying to see in different parts of the role of the plant, let's say, the role of either some molecules, coumarins in this case, that they might have in induced systemic resistance, or the communication with microbes. Let's say the project of SUPA is about coumarins in the microbiome root shoot axis. A project of Robin is how to develop next generation seed coatings using fungal mycelia. Then the project of Max is how do beneficial microbiota perceive coumarins. Then Alberto is studying the microbiome of ISR. Together with Ronnie and Gie, we study the role of root hairs in microbiome assembly. And the first part of my scientific talk is about the role of plant defense components and plant microbiome interactions. And this is work done mostly by Melissa and Xiaoyu. So I won't go to so many details, but at least I will try to give you some highlights. So this is an audience that is familiar with the microbiome, I expect. But what is really important to mention here is that the microbiome, that is this community of microbes and also the functions they encode that play roles in plant health and fitness, they are somehow selected by the plants. And we know this because there is a gradient. So if we go from the soil towards the root, there is selective selection process. And we know that because only a subset of microbes can, let's say, colonize the surface of the roots, but also the interior. And we know from many beautiful works that have been published in the last decade that a number of factors contribute to this chemical communication between plants and the microbes, such as primary metabolites. You can see here organic acid, amino acids, and sugars. Secondary metabolites like phytoalexins, triterpenes, benzoxazinoids. Also hormones. We heard about the role of ethylene in the previous talk. But I would also add here that also the structure of the root, but also the anatomy of the root could also contribute to how plants communicate with the microbiome. And what is really important to know is, let's say, how through this selection, plants can select beneficial microbes, because these beneficial microbes can help them with growth, promotion, nutrient acquisitions. They can enhance the resistance of plants. And one of these microbes that we study a lot at the University of Utrecht is Pseudomonas simiae WCS 417. So this is well studied for its ability to induce ISR, but also growth promotion. And in a previous study, we have demonstrated that some secondary metabolites produced in the roots of plants, coumarins, can really have a role in shaping the microbiome. And then we have demonstrated that some beneficial microbes seem to be resistant to these coumarins, while some pathogenic fungi seem to be susceptible. So then, since this is a published work, we want to expand this. And then, as you can see, coumarins are here. They're produced probably in the cortex, and then they are going out from the epidermis. But there are more defense components, chemical and structural, in the roots. So this is a cross-section of an Arabidopsis root. You see different cell types, okay, like epidermis, cortex, endodermis, and then the xylem. And then you see we have here, let's say, somehow painted with different colors, the localization of different chemical or structural defense components, like cutin, you see here is in the epidermis, the position of callose in the epidermis, coumarins, superdeposition in the endodermis, the position of lignin either in the xylem or in the Casparian strips that are in the endodermis, and so on. Or the production of glucosinolase and camalexin that is exported also from the interior part of the root in the rhizosphere. So to study now the contribution of these defense components, we made use of a set of mutants that are available, and this is work of Xiaoyu. We use these mutants, and initially we want to know what the role is of these different defense components in the interaction with a beneficial bacterium. For this, we grew the bacterium in a system where we apply the bacterium in the medium, and then we applied, we transferred the seedlings, either wild type or mutant seedlings, on top of this medium. And after 7 days, in this medium, we could not only assess growth parameters, but we could also assess colonization levels of the bacterium on the roots of wild type and mutant plants. Because this is a long story and a lot of graphs, we try to simplify the information. And what we found is, with blue, some of the mutants that had less beneficial response to 417 compared to Columbia 0, the wild type, and with red, more beneficial response than Columbia 0. And what we found is that in some of the traits that we assessed, like shoot weight, primary root length, and lateral root number, at least in the shoot weight and primary root length, we found a less beneficial response of some of the mutants to the wild-type plants. And then we selected some representative mutants, one that was involved in cuticle structure, one in Casparian strips, one glucosinolates, one camalexin, and then callose deposition, lipidification on glucosinolates. And we wanted to further know what exactly makes these mutants respond in a different way compared to wild type. Because this is, I don't have enough time in this presentation. I will just say that at the moment we have performed RNA-seq in plants, wild type and mutants that are colonized by 417, and we assess how these plants, the mutant plants, respond to the bacterium compared to wild type plants. But we have also exposed the bacterium to exudates coming from wild type, but also mutant plants, and we also assess how the bacterium responds to exudates coming from wild type and mutant plants. This is ongoing analysis. I hope in the next meeting I will be able to present more about this. But we don't study only the role of these defense components with one bacterium that is member of a more complex microbiome, but we also want to know what is the role of these defense components in the interaction with soil microbiota. This is work of Melissa. And to do this, we use the field soil and we grew the different mutants, and we assessed initially the shoot fresh weight. So what you can see here, we have wild type plants, and then we see that some of the mutants do not respond in this soil in a similar manner like wild type plants. So they grow smaller. That could be for many reasons. One of them could be the microbiome, and we decided to also study what is the microbiome assembly in these plants in different compartments, not only in the rhizosphere, but also on the rhizoplane, that is the surface of the roots, but also in the interior of these plants, the endosphere, because we don't know exactly where a defense component might have a role. If you consider that a mutation in a structural defense component could also affect exudation. Okay. So we assessed all these different compartments, and now I will just show you this slide just to get an idea of the work that Melissa is doing. And what we see here is if we go from the rhizosphere to the rhizoplane and the endosphere, we have a nice compartment effect in both chemical and structural defense barriers. Okay, so here we have grouped all of them together. And then we want to know which communities in which compartments are different compared to wild type plants. And actually what you see here, this is the rhizosphere, and with orange you see the communities that respond different compared to wild type. And in the rhizosphere we have some chemical defense mutants like coumarins and glucosinolates. On the rhizoplane we don't have only chemical but also some structural mutants like cutin and endodermal mutants. But if we go in the endosphere, then we see the strong contribution of endodermal mutants, but also of chemical mutants that, let's say, the compounds are produced internally, okay, in the inner cell types. This is ongoing analysis. Again, I hope to show you more in the next meeting. And this is one of the works we do in Utrecht, but I would like also to tell you some things about the work that Daphne is doing with me at the Agricultural University of Athens, where she's actually trying to find some, as we say here in a poetic manner, nature-based solutions to combat soil-borne pathogens. Actually, her work is about the interaction of tomato and the devastating soil-borne pathogen Verticillium daliae. And actually, what we try to do, we try to understand what makes a selection of wild relatives of tomato resistant to this pathogen. To understand that, we follow different approaches. One of them is to study whether the exudation of domesticated and wild tomatoes is different. Then we want to assess also whether anatomical defenses of these wild relatives is different compared to the domesticated partners. And we also want to profile the microbiomes in this case to see if these wild relatives assemble a different microbiome that could make them also more resistant. And some preliminary findings of Daphne I can show you here. So what Daphne has done, she collected soil from an agricultural field, and then she compared the behavior of the domesticated cultivar compared to a soil substrate that we usually use in the greenhouse, but also compared to autoclaved field soil. So what you actually see here is the percentage of wilting, because one of the symptoms that verticillium is causing to tomato is wilting. Okay, and what we see actually is when the plants grow in field soil where there is a rich microbiome, they have less wilting compared to plants that grow either in autoclaved field soil or soil substrate that has a low microbial community in terms of diversity and composition. Okay, and the next thing that Daphne did, she compared the behavior and the fitness, but also the resistance of the wild relatives compared to moneymaker. This is the domesticated cultivar we use. And what she assessed was the height. So you see here that the wild relatives grow taller, but are also more resistant to the pathogen compared to the domesticated partner. And this is also ongoing work. So now what we do, we are analyzing exudates that we collected from moneymaker and wild relatives in collaboration with the University of Tusa in Italy. And with this, I hope you got an idea of where I work now, some of the things that we do in the lab, and I think I gave you some highlights of my work, both at the University of Utrecht, but also at the Agricultural University of Athens. And with this, I would like to thank you for your attention.
Okay. Your turn, Naomi.
Yes. Okay, yeah. So very much thank you, Giannis, for a very interesting presentation and for providing such a nice overview of the University of Athens as well. It looks like a very nice place to go. Yeah, then without further ado, I think we can go to questions. Are there any questions for Giannis?
Hi, yes, I have a question for Giannis. So thank you, Giannis, for the nice talk and for the nice overview of your research. I have a question related with the exudation of plants. I mean, are you also playing with the stress conditions? Because we know that under such conditions are where plant interacts with other microbes, right? Pathogens or even beneficials. But so are you playing at the time for taking the exudates? Are you playing with the nutritional or I don't know, drought stress or salinity, etc.?
So thank you for the question. Unfortunately, I cannot see you. So previously, at least with the work with coumarins, yes, we worked with nutrient deficiency, in this case, iron deficiency. And now in the work that we do with Daphne, the stress is the infection by the pathogen. So we analyze exudates, not only from plants that grow without infection, let's say healthy plants, but also plants that are infected by the pathogen. Because we also want to understand the dynamics, let's say how the plant also responds to that and how that could shape both the interaction with the pathogen. So in the next step, we want to see which exudates we could use against the pathogen. One thing is this, but also if we see differences in microbiome assembly, we could also make some correlations, like there is presence of this class of exudates here that we don't find in this condition that could explain why we have specific recruitment of microbes.
Okay, but not playing with abiotic stresses. I mean, because we know that plant exudation is changing because of the nutrients or other kind of stresses, and producing signals as a cry for help, right? Yes. So if everything is optimal, again, you are playing with the pathogens, so this is not optimal for plants. So I understand that they also may change in the exudation pattern. But yeah, very nice, and we should talk definitely. Yeah.
And,
thank you for the nice talk.
Thanks a lot. And just to add on this, in micro SOS, actually one of the stresses we have is drought, and another stress is salinity. And there we also want to study how to use microbes to help plants deal with this. And we are going to also analyze metabolomes and so on.
Nice. So, thank you.
Thank you.
Giannis, I think you have a question in the chat about Elisa Lu. Low.
Okay,
No, they're not pronounced properly.
let me see. Okay.
Can you see? Oh, I can read the question for you. Or you see the question.
Yes, I see the question. Thank you for the question, Elisa. I have to say that it's indeed interesting, but we have seen in the past that when the plants are stressed, they might also produce more lateral roots, and that's a way also to avoid the stress. But I have to say we haven't really elaborated on this. This is something that we also check now in the RNA-seq datasets to see if we have some leads that could explain this differential growth response. But it's actually something that we also observed, and it was interesting.
I don't know if there are more questions. So I have a very silly question, probably, but so the mutant that you see that they have the radioactive mutant that they have different growth in soil. How are they in vitro conditions? So are they similar to the wild type? They are smaller, because just to see if they have a penalty at the beginning or not.
So we have seen that, let's say, these differences between wild type and mutants are way stronger when we haven't autoclaved the soil. So when we have a soil that's gamma irradiated, you see that these differences are not so strong.
Okay.
That's why we think that microbiome is a main factor, among other things, because of course they are mutants. They could have also other defects in acquiring nutrients and so on. But one of the things that we did, because we have grown them also in vitro, we haven't seen major differences in vitro, but also in soil that's gamma irradiated. And we see only when we have microbiome present.
And related with that, have you tried, I know it's difficult, but different type of soils? Because sometimes just to see you have different, I mean, I know just with one soil is completely, it's already difficult, but just, I mean, it's a curiosity.
Short question, a short answer, no. Another short answer, we have thought about it also. But the answers are short, the experiments are long. So, no.
Okay. I completely understand.
No, but it's a totally logical thing, because you observe something in a soil, you want to know if you observe it also in another soil.
Yeah.
That could be done probably later, once we find some really interesting leads.
Okay. So I don't know if there are more questions. If not, thank you, Giannis, again. It was really interesting.
Thanks,
and I will now we will refresh the next speakers again. So let me check 1 second.
Yeah, so thank you again to both of the speakers for a very nice webinar, I would say. It was very informative. And then, yeah, indeed, I'm waiting a bit now for the fan to
Yeah.
Didn't have to wait very long. So indeed, as I said, it's bi-monthly, so every 2 months. So the next session will be at the 30th of May. And there we have the pleasure of listening to Professor Uta Paskowski as well as Professor Enrique Monte. So make sure to join us next time.
Yeah.
And thank you all so much for joining as well.
Okay, so I think that's all. So have a very nice weekend and see you soon, some of you in Germany.
Yes.